Dr. James Duwe, welcome so much to Norway in a digital way, but in very much a heartfelt way. Welcome to you.
Thank you.
Well, a lot of people in Norway, we don't know you yet, but I love your YouTube channel and I love how you think because, a lot of people have theories, but you try out the stuff, you do the actual science. And I've actually got 4 kids and I always try to teach them reality is a good test for truth. And I think that's what you do. And so I really appreciate it. But for people to get to know you a little bit, what is your scientific background?
I have a PhD in chemistry, but I work across many fields. I've been a professor for over 30 years, and we work in the area of everything from medicine to material science to electronics. And to the extent that I've published lots of papers in all of those fields, as well.
How many papers is that roundabout?
It's over 700. I don't remember the exact number, but it's over
How did
700.
you do that?
Oh, by the grace of God. We just take one at a time. But I have some tremendous students, and we just keep putting it out there, and they keep doing wonderful work. But if I could really give glory to God in this, I'm not a particularly high IQ person. I was never the strongest student in the class. I don't work as a minister in the formal sense. I work as a chemist. So I just pray that I get filled with real understanding and insight. And we've been really blessed. I have 14 companies that have spun off from my work. And so it really does hit the practical level as well. And we've just been greatly blessed.
But I also very much appreciate you putting that focus. And at the same time, you're saying that you're not just thinking theoretically about science, but you're doing science, you're making inventions. And I love that part. So we're going to look at a little bit at some biology textbooks here in Norway. And I would love to read you something. And it starts with the small stuff. So it's about the beginning of life. And I know you like building small stuff. I think you build nano cars where you can have, is it 50,000 on the diameter of a hair?
Correct.
And you won a race last year too, I think?
In 2017.
2017?
Yeah. And there's race number 2 is supposed to be in 2022.
Good stuff.
Yeah.
I didn't even know they raced nano cars until I saw that on YouTube, but that sounds exciting. So this first quote, it's about how life first arose. And it says, how life first arose, we may never get an answer to. Very fair. But scientists think it happened like this. Simple molecules got together and made complicated molecules, maybe through lightning bolts or through sun rays, since there was no ozone layer. The process repeated itself and produced still more complicated molecules that made chains. And after millions of years, they organized themselves together in order to be able to eat and digest food. And then they were able to divide, and the border had been crossed and life had evolved. It's just a theory, but scientists say it happened in the oceans. And just shortly, a second one about the same thing, which gives a little bit more details. Under specific circumstances, a prebiotic soup can organize itself and make a protobiont of polymerase and organic stuff. A protobiont has a structure similar to a cell, but no organs or membrane, just a thin skin to be protected against the outside world. So basically, we're reading how simple molecules, step by step, and through chemistry, which is your field, became life. And I was going to ask you, given enough time, does this seem possible?
No. None of those steps seem possible. The books are accurate in that is what many scientists say, but there is no evidence for any of those steps that are suggested there. The small molecules coming together to make larger molecules that are of biological interest, it's very hard to see how that could happen on a prebiotic Earth. If it happens in the oceans like what was suggested, the concentrations would be way too low. In other words, there would be other reactions occurring before those reactions could ever take place in the first place. Then there would be the other problem is that their degradation, in other words, just the reaction with water and with salt in the water, would be much faster than the other reactions that you need to occur with them. So just the kinetics, meaning the reaction rates, don't work. Once you get these biologically interesting molecules, for them to hook up is very, very hard to imagine. They don't just hook up by themselves. They actually go back. They depolymerize by themselves in the ocean. So water has a tendency to hydrolyze them. So it goes the other way. The molecules that would be needed all have to be what are called homochiral, have one particular chirality. And so what happens is without that, you don't get the reaction rates to occur in the same way. You don't get the reactions to occur. They generate too much heat. And that's something that I talked about in my series called chiral-induced spin selectivity. As far as the protobiont, the structure that protects it from the outside world, if it's protecting it from the outside world, then the nutrients can never get in. And so you have to have selective ports to let these things in. No human-made protobiont in the sense of trying to do something that mimics early Earth has ever had that kind of constituency. You have to have what's called a proton gradient, a different level of protons, different level of acidity on the inside and the outside, if you will. By acidity, I'm talking about the amount of protons on the inside and the outside. That has to be extremely well controlled, but you have to have certain proteins or things that would open up in that lipid bilayer to allow it. Even the lipid bilayer itself of the protobiont has to have a non-symmetric arrangement, meaning that the outer layer has to be different than the constituency of the inner layer, or else you don't get this proton gradient. And so all of these different pieces make it impossible for me to think how that could occur. And what's interesting, as soon as you start talking about the exact chemistry that's needed, even the scientists who would say, yes, that's how it's done, it's very hard for them to see it working either. That's not a bad hypothesis in the sense that that's the best we've got. Now, some people will throw up their hands and say, well, it just came here from outer space. That just does what's called begging the question, meaning that you have to just say, well, where in outer space did it come from? If some alien planted it here, we're talking about not the origin of life just on Earth, the origin of first life, period. And we have to say, okay, well, where did it occur? How did it occur? So just saying it came from outer space doesn't solve the problem for us.
Oh, you're just moving it basically. And
correct.
I know this quote that you have sometimes by Richard Feynman. He says, "What I cannot create, I do not understand." And that basically links in very well because you've tried some of this stuff. But I'm going to read you another quote. I can't give up on these Norwegian textbooks so easily, you know. Sure. It says, molecules turned into RNA. Let's suppose they were these molecules containing digital information. And then it says, it's reasonable to assume that RNA managed to run simple forms of life without DNA. Tests in a lab have shown that RNA can make copies of itself as an enzyme. And this RNA enzyme or machine can put nucleotides together, and natural selection has been observed in this so-called RNA world. So basically it's saying there was this RNA world and it was simpler, and that's kind of what made it work. And did that RNA world solve the puzzle of life?
Right. So the problem with making the RNA to begin with is you have to have the nucleobase, which is not that hard to make. But you have to be able to hook that onto what's called a ribose sugar, which is very hard to make. And if you did make it, it's very hard to think about how that was made in homochiral form. The problem then with RNA is if you were to be able to get it to polymerize, and people have tried to get RNA to polymerize, it's very difficult under prebiotic conditions without enzymes around. So if you were able to get it to polymerize, its stability is very low, even on the order of hours at room temperature, even on the order of minutes, and its decomposition is catalyzed by metal ions, of which there's metal ions all the time around in oceans and in water. So in other words, when we stabilize it, we add what are called metal chelators to try to grab all the metals in there. And RNA is often stored at minus 80 degrees centigrade. Minus 80 degrees centigrade because of its instability, but that alone would not stabilize it. You have to have chelating agents to make sure there's no metals that cause it to decompose. So it has to stay very stable. So if you're going to get up to reaction temperatures where reactions can occur, it's decomposing generally very quickly. As far as RNA copying itself, it's never been known to copy itself except very small portions of itself in primed. That means human-designed RNA that is primed, that's ready to copy itself. It's only been able to copy about 10% of itself and then stop. So you only get something about 10% as long as what the RNA was. So that's not a copy. And that new 10% is not long enough to be a template for another one to be copied. So even this suggestion that it copies itself is very hard to fathom. With people trying under the best of conditions, it's very hard to fathom. The other problem is when it does copy itself, the two RNA molecules stick together very strongly. They don't come apart. So if you actually read in the experiments where they do things, where they suggest, for example, RNA was made on a clay template or something, you have to add what's called ion exchange resins. You have to deal with the ions to get them apart. So the problem with RNA is it has this too sticky problem. It sticks to itself too much. And RNA has never been shown to do the very things that you're talking about. So RNA is the system that takes amino acids and hooks it together to make enzymes to build cellular components. But without other associated enzymes which help to bring in these components, it doesn't work. So there's so many pieces that would have to come together at once. Again, it's very easy to throw this out as a suggestive idea, but as soon as you get into the details, it becomes very hard to figure out.
Yeah, and I'm happy that you know the details, so we can look at it together. For my work, actually, I'm a cabinet maker. That's my trade, to build furniture.
That's like Jesus.
Yeah, he built all kinds of stuff actually when the word technon. So yeah, I love. I have a little farm. We got some sheep and some chickens, and I build stuff with my kids. And you have to come to Norway to visit one day. But I love making stuff. And I don't know if you've read Signature in the Cell by Stephen Meyer, but he somehow explains a little bit what needs to come together when it comes to the DNA. And so this next textbook passage, it says, to get from RNA to DNA and proteins, the genetic code needs to be evolved. RNA is single stringed and cannot be corrected, so it can only be short chains. DNA is double stringed and can be corrected, so you can get bigger genomes and I'm guessing, build more complicated stuff. But it doesn't actually tell us how to get from RNA to DNA. Can you do that? How does that work?
Okay, so one of the things that I didn't address in the last section that you brought back up again is the informational code. So just hooking together RNA with its different nucleobases does not give you the type of code that you need to prescribe the synthesis of any useful enzyme. And there's different kinds of information. There's an information that can come from randomness, which is called Shannon-like information. That's not what we need. We need what's called specified information to be able to do this. And that's never been realized. And where that comes from, we just don't know. Can RNA act as a template for DNA synthesis? The answer is yes, it can. Because you can get alignments and it can act as a template for DNA synthesis. The problem now becomes that, again, but to make DNA, you have to have the RNA where you're working off of ribose. You have to have DNA where you're working off of deoxyribose. You have to have RNA that has a certain type of bases that it uses. DNA uses similar bases on 3 of them, but has a different letter. So each of them have 4 letters, but only 3 of them are the same. There's a unique letter on each of the other. How those would distinguish each other, we just don't know. It could act as a template, but what would happen is if it ever did form, you would have an RNA DNA duplex, which again is really quite sticky, and they would stick together, and you have to have all the homochirality right, or the thing just doesn't work. And the problem with the RNA is you have 2 possible linkages. You have going to the 3' or versus off the 2' center, and that breaks up the chain immediately, and it's a real problem. So yes, you could say that RNA might make DNA, but that doesn't get you anywhere, in the sense that you already have the RNA. Where did the information come from in that RNA? If you were to say it came about randomly, well, then the probabilities become really, really low. Just for one RNA, and that RNA prescribes for a particular protein sequence. But what happens when you need another enzyme? Because you can't build a cell with just one enzyme. So you have to have at least, it's been calculated, you have to have at least 206 protein coding genes to be able to do this. So you have to have at least 206 different specified areas to make the minimally controlled system. And that would be a cell that would have to have all the exogenous amino acids come in exogenously from the outside, because it doesn't have the capability to make its own at that level. So it's very hard. The number of components is so difficult. If I could put it in relation that okay, you could have the block of an engine. You could have the block of an engine, but somehow you've got to get the pistons with the piston rings inserted into that. You've got to find the pistons. You've got to put piston rings. You've got to get those inserted. You've got to hook all that up to the camshaft, and you've got to have all that hooked up to the ports being put in right. You have to have the spark plugs to be put in right. There's so many different components. So to just say that you have an engine block and maybe somehow once in a while a piston slipped in, you know how hard it is to get a piston in when it has a piston ring around it. But somehow, then you have to do this over and over and over again. But there's so many other parts that have to be put together. It's like saying you could put all those together in a box and shake it together and they fall together. It's very hard to figure out how that's done. And even if they all came together, that engine has to be hooked up to many other pieces to begin to operate. So it's like that problem, but orders of magnitude more difficult.
You know, with this kind of background, it becomes very improbable, but somehow people just have this time thing, you know, like if you just get all the parts together in the same area and somehow by luck and chance you get a membrane around it, and then that's, you know, lucky things do happen. So people say if you just add time, why couldn't they be in one place on Earth? Why couldn't we just get lucky, you know? What would you say to that time factor?
Right. So time turns out to be the enemy when you're dealing with organic compounds. Like you mentioned, an early Earth didn't have an ozone layer. If that's the case, you have a lot of UV radiation coming in. UV degrades organic compounds very quickly, actually. So let's not go without the ozone layer, or else we couldn't even get started.
Okay.
But so you'd have UV rays coming in. You have lots of oxidative processes coming in. If you say the early Earth was reductive, so now you have a lot of reductive processes coming in, very high in ammonia. Ammonia can be extremely degradative to organic compounds. And so you see that the chemistry now becomes very, very difficult still. And time actually hurts things, because usually for the types of biological molecules that we want, which are of interest, their degradation is faster than their formation. The degradation is faster than formation. It's not just me saying this. I was, in fact, seeing a recent video by Jack Szostak, which is a main proponent on the origin of life, a Nobel Prize winner from Harvard and Harvard Medical School. And he was even saying that. There was somebody said, well, we have plenty of time. We don't even need catalysis. And he right away stopped them. He says, no, the degradation is faster than the synthesis, number one. And then during his talk, he showed the number two side, which is that even hot-cold cycles. People will say, well, it gets hot and cold. Even cold cycles between day and night is not fast enough to get these things polymerized, because the degradation rate would occur during the non-synthetic cycles. That these things are only stable on the order of minutes, So it's a big problem. Time is actually an enemy in this whole thing. It's like if you leave your car parts out in the rain, in the weather, and you say, well, eventually these car parts, there'll be some earthquakes and they'll kind of shake together. The degradation before they ever get together is much faster than their assembly. That's the problem.
I understand that because I love cars. I got Mercedes 1963 truck that I used to get manure to my farm, and I got 2 or 3 old motorbikes. Yeah.
Interesting. I'd like to use the analogy as well, because I used to work on cars a lot growing up, a lot on cars and motorcycles. I worked in a repair in a gas station for 4 years in high school, and I drove a service truck up and down the highways outside New York City, and to go and try to fix a car on the side of the highway. So it wasn't just in a pristine shop where I had to do this. I had to do this often by the side of the road, and you use whatever parts you have to get them to move along.
And it didn't happen by itself.
It didn't happen by itself. They had to call somebody to help them.
Yeah.
And if they waited, it would not have solved the problem.
No. And sometimes I think it's a little bit like sometimes people say, if you throw the dice long enough, you get the numbers. But the dice, they're really made of ice, and they're disappearing as you're throwing them.
Yes.
Which is kind
that's
of
a good analogy.
Same thinking. So what's your basic conclusion, if we want to close off this first session? What's your basic conclusion about origin of life science?
Well, it's as I say, we're clueless. We just don't know. So we do not know the origin of life, even if we had all the components. I mean, you have all the components. You have a living cell. You have all the components. If I gave you all the RNA, all the DNA in the sequence they wanted, so it has the information. If I gave them all the lipids, all the saccharides in the sequence they wanted, and I say assemble a cell, you'd get a Nobel Prize for that if you could do that. Why hasn't somebody done it? If the hard part is getting the components, I'll give you the components. Now just try the assembly part in your pristine laboratory. Do what you got to do. Knock yourself out. You won't even get close. We see this all the time. A cell just dies. A cell just dies, and it just stops functioning. How do we get it started again? I mean, most everything is in place. How do we even get it started again? It's hard to think about that. And the alignment of all the non-covalent alignment, it's just so utterly complex. And the problem, Chris, is that the target that we're trying to get at, a cell, gets harder every year. And it's not because the cell evolves and gets more complex. It's just that our knowledge of the cell says, wow, this is a lot more complex than I thought it was. And then the next year becomes, wow, a lot more complex than I thought it was, because we learn more about the cell. So our target is moving further and further away, much faster than we're able to approach it. So we're actually getting further away from solving this problem. Further away.
The more we know. Yeah.
So we just don't know. Now, I could never claim that we'll never know. I can't say that. As a scientist, I could never say we'll never know. I just say that we're nowhere close today. And it's not going to happen in the next year or the next year or the next year, because the target is moving away much faster than we're moving toward it. There's so many hurdles that have to be solved. So I don't know when this is going to be solved. I don't know if it's going to be solved, but I could never say that it cannot be solved.
All right. Well, to me, it sounds a little bit like we need something more, but I think we can talk about that in the next session. Thank you very much.
Thank you.